Okay, so for the forward and the reverse grouping with the ABO blood group testing, you have to set up your forward and your reverse at the same time. So when you do your forward testing, the reagents we're going to use are the antisera, like the known antisera. So the types of antisera that you guys know that we use are anti-A and anti-B. So those are the two that we can use. We can also use anti-AB, which is a third one which contains both anti-A and anti-B in it, and it's just a confirmatory test. But we don't use that every time, so I won't bother just for this quick explanation. And then for the reverse grouping, we're going to use reagent known red blood cells. So they're going to have antigens on them that we already know. So the two that we use for that are the A1 cells. So they have A1 antigens on them, and B cells. And this is going to be standard for no matter what patient we use. So that's the basic setup. So you're going to have a tube set up. I'll use a different color. You're going to set up a tube for this one, here, here, and here. And so you have to have 4 different tubes. And you have to put the anti-A in this tube, the first one, anti-B in this one, A1 cells in this one, and B cells in this one. So you can't put them all into one tube. Each test has to be done separately. So if we have antisera with antibodies, and they're known, and they're going into into these 2 tubes here, that's our known reagent. So if antisera is going in, that means we need to put red cells in to test. So if the patient is group A, we're taking we have their EDTA tube. This is going to be the same for everybody. We have their EDTA tube. So we spin it down and we have our red cells on the bottom for the patient. And then we have the plasma on the top. And you guys know now that like the plasma has the antibodies in it and the red cells have the antigens on them. So this is our patient and they're unknown. We don't know what their blood type is. We know that they have red cells and we know that they have plasma. And depending on their blood type, they could have the naturally occurring antibodies in their plasma. But we don't know yet, so we have to figure that out. So if the patient was group A, which we wouldn't know until we tested it, but this is the group A example, we're going to take their patient red cells and put them into here. So we take, they're not packed cells. We have to make a 3 to 5% cell suspension. And you guys know about that. We've talked about that before. So it's the cell suspension that goes in here, and it's just 1 drop. And then we take 1 drop of the known anti-A. So I'll do that in green here. So add your anti-A, and now we're adding known antibodies into this tube. And then we do the same thing in a separate tube for B. We take that patient's 3 to 5% cell suspension, and we put 1 drop in here, and they're going to have antigens. These 2 tubes have antigens on them. We just don't know which ones they are yet. And then we add our anti-B, known antisera, to this tube, which is antibodies. And this is anti-A and anti-B, you guys know, was IgM. And IgM reacts at room temp. So it just needs an immediate spin. That's all we need to do. We don't need to incubate it or anything. IgM reacts at room temperature. So we're just going to do a 30-second spin. And it's going to mix everything together. And we know that brings the antibodies close to the antigens. And then you're going to shake your tube and you're going to observe for agglutination. So with a group A person, I'm going to erase this other stuff here. If they're group A, we're going based on what's on their red cell surface. So if they're group A, we know that this is anti-A and it's going to react with A antigens on the red cell surface. So after a 30-second spin, this is going to be a 4+ agglutination. You're going to shake the tube and you're going to see a 4+ agglutination. And again, if they're group A and you add anti-B to the red cells, are you going to see any agglutination? No, that'll be a negative. So you'll shake your tube and it's going to go back into suspension. These anti-B antibodies are not going to agglutinate these red cells because there aren't any B antigens on there. There's A antigens, and that gets picked up in this one. So if a patient's group A, that means they have A antigens on the red cell surface, and the anti-A reagent will detect those and agglutinate them, and you'll see a 4+. And then you'll see a negative reaction with the anti-B because there are no B antigens on a group A individual. And then to confirm that, at the same time, you're going to take the patient's plasma over here. I'll use red or blue. You'll take the patient's plasma and you're going to add it to your A1 cells. So you'll have reagent A1 cells, which we know are going to have little A1 antigens on them. And we're going to take the patient's plasma with antibodies and we're going to put them into the A1 cells. So now we have patient antibodies, and these are the naturally occurring IgM antibodies. So in this example, in the forward, the antibodies were reagents. In this example with the reverse, the antibodies are from the patient. So you have to add the antibodies to your A1 cells, and then you're going to add your antibodies to your B cells in a different tube. And they have little B antigens already on them. We know that because they're reagents. So now, if the patient is group A, their forward grouping is going to have this reaction, a 4+ in the anti-A and a negative in the anti-B. And now when we take their plasma, we want to confirm what their forward grouping is saying. So if we take patient plasma from a group A individual, you guys know that they have the opposite antibody naturally occurring in their plasma. So if they have A antigens on the red cell surface, they're going to have anti-B in their plasma. So if we know that, if we add their plasma to this tube with A1 cells, these antibodies should be anti-B. They should be the opposite of their blood type over on this side from the forward. So if we're assuming that they're group A, these should be anti-B antibodies. So we should not see a reaction here. Those are A1 cells. They don't have B antigens on them, and these are supposed to be anti-B. So this is going to be a negative reaction. The anti-B in their plasma has nothing to agglutinate here because these are all just A1 antigens. So this, you'll shake the tube, or you'll spin it down for 30 seconds, an immediate spin for 30 seconds. And then you'll shake your tube, shake off the button, and this will go back into suspension because there's A1 antigens on there, and there's nothing for the anti-B to react with. But if we look at our B cell tube over here, like I said, we know that these are anti-B antibodies because that's what the forward grouping told us. The patient was typing as group A, so they should have anti-B antibodies in their plasma, and that's what we added here. So now the anti-B does have something to react to. These are B reagent cells with B antigens on them. So the anti-B in their plasma is going to agglutinate those B reagent cells, and you should shake your tube and see a 4+ reaction there. And now that makes sense with what the blood type is. So we have a 4+ in the anti-A, a negative in the anti-B, and then you should have a negative in the A1 cells and a positive in the B cells. And this is a straightforward right here. This reaction is the standard for a group A person. 4+, 0, 0, 4+. So you guys can memorize that, but it's not going to do you any good. Like if you memorize a group A person has this reaction pattern, that's fine. But if you were to try to explain why this reaction pattern occurred, you won't be able to if you just memorized it. You have to understand that you took the patient's red cells and you added them to the anti-A, and you should see agglutination there if they're group A. And then you should not see any agglutination in anti-B because there aren't any B antigens there. And then we look at the reverse, and it's the same principle. We add their plasma, and they have the opposite antibody in their plasma to what they have on their red cells for their antigen. So their anti-B should not react with A1 cells, but you will see it react with the B cells.